The overall goal of this procedure is to evaluate the phenotype and suppressive function of natural CD4-positive regulatory T cells expanded from the peripheral blood of HIV-1-positive individuals. This is accomplished by first isolating the regulatory T cells by density gradient and flow-based cell sorting. In the second step, the regulatory T cells are expanded in culture. Next, the phenotype of the expanded cells is characterized by flow cytometry. In the final step, the function of the expanded Treg population is assessed by a T cell suppression assay.
Investigating natural CD4-positive Tregs in HIV-infected individuals has traditionally been challenging, and in large part because of the scarcity of the population. The advantage of our technique is that it allows for the ex vivo generation of large numbers of these very rare cells.
The method demonstrated here may help to answer key questions in the HIV immunopathogenesis field by facilitating the characterization of the function and specificity of regulatory T cells in HIV disease progression.
Though this method can provide insight into peripheral blood regulatory T cells, it can also be applied to other body compartments, such as expansion of regulatory T cells from the gut-associated lymphoid tissue, a major site of HIV replication.
Begin by carefully transferring the blood from the ACD collection tubes into 50 milliliter conical tubes for a final volume of 15 milliliters of blood per tube. Then add 25 microliters of Rosette-Sepp human CD4-positive T-cell enrichment cocktail per milliliter of blood, mix carefully, and incubate the cell suspensions for 20 minutes at room temperature. After the incubation, carefully mix 15 milliliters of PBS supplemented with 2% FBS with the blood. Then layer the diluted blood sample on top of 15 milliliters of histopac in a 50 milliliter conical tube. Spin the blood for 20 minutes at 1200 Gs and room temperature with a slow start and no breaks, and then transfer the CD4-positive T-cell enriched PBMC layer into a new 50 milliliter conical tube. Wash the cells in PBS supplemented with FBS, count the cells, and then, after another wash, resuspend the pellet at about 20 × 10 to the 6 cells per 200 microliters. Add the following antibodies at the indicated concentrations, and then incubate the cells for 30 minutes in the dark at 4 degrees Celsius. After washing away the unbound antibody, resuspend the cells at 20 × 10 to the 6 cells per milliliter in additional PBS plus FBS, and filter the cell suspension through a 35 micrometer nylon mesh. Now, using a cell sorter equipped for handling biohazardous material, sort the CD3 positive, CD4 positive, CD25 positive, CD127 low Treg in ex vivo 15 media using this gating strategy. Conventional CD3 positive, CD4 positive, CD25 negative, CD127 positive T cells can then be isolated and expanded as negative controls. After sorting, wash the Treg cells with ex vivo 15 media and resuspend the pellet at 250 × 10 to the 3 cells per milliliter in ex vivo 15 media, supplemented with human serum, penicillin, and streptomycin. Then, after washing the human T activator CD3-CD28 beads according to the manufacturer's instructions, add the beads to the isolated Treg cells at a ratio of 1:1 bead per cell. Then, after 2 days of culture, double the media volume and add IL-2 to the cells. Now culture the Treg cells for 2 weeks, changing the media on days 5, 7, 9, and 12. On day 9, add beads to the culture at a 1:1 ratio. At the end of the culture expansion, wash the harvested conventional and regulatory T cells in PBS. Then use the live dead fixable violet dead cell stain kit according to the manufacturer's protocol to stain the dead cells. Wash away the excess stain in PBS and FBS. Next, add the following antibodies at the indicated concentrations and incubate the cells at 30 minutes in the dark at 4 degrees Celsius. After another wash, stain the cells with a FOXP3 transcription factor staining buffer set according to the manufacturer's instructions. then add the following antibodies at the indicated concentrations, and acquire the data on a flow cytometer. Also at the end of the expansion culture, thaw autologous cryopreserved ex vivo PBMCs, rest the cells for about 3 hours in a 37°C incubator in complete media. After staining dead cells as just demonstrated, wash the cells in PBS with FPS, and then incubate the cells with anti-CD3 PE-Cy7 for 30 minutes in the dark at 4°C. After washing the cells again, resuspend the pellet in PBS and FBS and filter the cell suspension through a 35 micrometer nylon mesh. Then use a cell sorter equipped for handling biohazardous material to sort the viable CD3 positive T cells into R10 media. After sorting, label the cells with CFSE according to the manufacturer's instructions and resuspend the cells in R+ media supplemented with human serum at 1 × 10⁶ cells per milliliter. Now harvest the expanded Treg cells and resuspend them at 0.5 × 10⁶ cells per milliliter in human R10 media. Then further split the cell culture into 0.25 × 10⁶ cells per milliliter and 0.125 × 10⁶ cells per milliliter dilutions. Next, resuspend anti-CD2, anti-CD3, anti-CD28 microbeads prepared according to the manufacturer's instructions at 0.75 × 10⁶ beads per milliliter in human R10 media. Then further dilute the beads with more R10 media to 3 different final concentrations of 0.625, 0.562, and 0.5 × 10⁶ beads per milliliter respectively. Culture the diluted cell and bead suspensions in a 96-well round bottom plate according to this protocol. After 4 days of culture, wash the cells and then incubate them for 30 minutes at 4°C with these antibodies. Finally, acquire the data on a flow cytometer. This first figure represents the gating strategy used to flow sort single CD3 positive, CD4 positive, CD25 positive, CD127 low Tregs from PBMC isolated from an HIV-1 positive individual. The anti-CD25 and anti-CD127 antibody clones and their conjugates are crucial for good separation of the Treg population. In this representative example, the Treg expansion reached a 765-fold change after 14 days of culture. The expansion fold change can vary from one individual to another and can range from 100 times to 2,500 times above the starting population. In this figure, the gray histograms represent staining for selected Treg markers, for example, CTLA4, FOXP3, and Helios. A fundamental characteristic of regulatory T cells is their capacity to suppress T cell effector function. Using a standardized flow-based T cell proliferation assay, this figure shows how regulatory T cells can suppress the proliferation of activated T cells at different T cell to Treg ratios.
After watching this video, you should have a good understanding of how to isolate and expand functional Tregs from HIV-positive individuals.
While attempting this procedure, it's important to remember to apply stringent gating criteria for sorting of the Tregs, as conventional T cells will also be expanded and may contaminate and overgrow the cell culture.
Following this procedure, other studies like comparing the function or gene expression of fluorescent HIV in vitro infected versus uninfected Tregs can be performed.
Don't forget that working with HIV-positive specimens can be hazardous and that precautions, such as following strict blood-borne pathogen standard operating procedures and working with a cell sorter equipped to handle biohazardous material, should always be taken while performing this procedure.